Hey guys, welcome to the explanation video for the week 14 PARTE Packet. In this video I will go over the answers to all of those questions, and this packet deals with biomolecules, so everything from proteins to DNA to different functional groups that we find often in biological molecules. So we'll get started. The first question, actually the first several questions, deal with proteins, and proteins are polymers made up of the monomer and amino acid. So there are 20 different amino acids in your body, but they all have the same basic structure where they have a carboxylic acid on one side and then this will be joined by a central carbon with what we call an R group or a variable group. This is called the side chain of the amino acid. So all 20 amino acids will have a different R group, and that'll determine the properties of the amino acid. And then on the other side we have an amine. So we call it an amino acid because we have an amino group with a carboxylic acid. So if you'll notice, we have two different reactive groups on the two sides. So we have this acid on one side, and then we have an amine on the other side. And when we react these two together, these two will be your two reactive groups, and it will combine with another one. So I'll draw another one in blue. And I'll do this one kind of upside down. Here's our carboxylic acid. Here's our R group. This one could be the same or it could be different. We don't know. It just depends on the amino acid that we use. And there's the amine group. So these two are going to react. And I will draw what comes from the black one in black and then the blue one in blue. So hopefully that kind of makes sense. We have the carboxylic acid that's still the same. We have the R group that's still the same. And then our nitrogen and only one of the H's, because one of these H's is actually lost. This is the linking bond that we just formed in red. You'll remember that's called an amide bond. And then here's the carboxylic acid of the other side, which is now just the carbonyl, or the double bonded O. So this whole group here is called an amide. Then here's that R group, and then here's the amine on the other side. And we have the same two groups on the opposite sides, so we could keep doing this reaction. That's why it's a polymerization reaction, because we can keep going and keep making this polymer longer. So what we've also done is we've gotten rid of this OH. So that means that we've made an HOH molecule, which you might recognize as H2O. So what we've done is we've taken out water, or we've condensed out water. So looking at this reaction, you know that we had two reactive groups and we got rid of something. So that one is going to be a condensation reaction. So our next one relates pretty closely. What two functional groups present in amino acids react when a peptide bond is formed? So this is our peptide bond. And the two groups that reacted were the amine and the carboxylic acid. So our answer here is also going to be D. So the next one, of the amino acids seen below, which highlighted side chain will be can be classified as polar? So if you remember from your electronegativities, the electronegativity of carbon and hydrogen are very similar. So when you have a C to H or a C to C bond, those will not be polar. But when we have something like a C to O or an O to H, those are going to be polar. So let's just look through these and see if any of these can be classified as polar. So this side chain is just a carbon. So this one is not going to be polar because we don't have any polar bonds. This one is also only made up of carbon and hydrogens. So this one will not be. This one we have an alcohol. So just drawing a little bit of this. Our oxygen is going to be much more electronegative than either of these other two things that it's bonded to. So this will be polar here. So, and then these other two examples, we just have carbons and hydrogens. So these are non-polar. And even though this benzene looks a little tricky, because we have double bonds, but double bonds don't make anything polar if we don't have more or less electronegative atoms, because these are still just carbons with hydrogens. So the answer here is C. All right. So of the amino acids seen below, which side chain would have the strongest intermolecular interaction with water? So let's think about our intermolecular forces that we learned. The strongest one is going to be electrostatic. So something where you have a positive charge and a negative charge. So that's electrostatic. Then after that, or that can also be called ion or ionic. The next one that we have is going to be an ion with a dipole. And then after that we have dipole, dipole, and then we have induced dipole, to dipole, and then finally we have the van der Waals, which are just induced dipole, induced dipole. Oops. Did the W first. Okay, so water, we know, isn't charged because it's not an ion, but it does have a dipole because of these two bonds. So the net dipole is like this. So the only interactions that we could be looking at are these guys, because we only have one ion, or we don't have an ion with water, so the most number of ions that we could have is one. And then it's not van der Waals because we don't have a nonpolar molecule. We have at least one polar molecule because we have water. So one has to be dipole. So let's look at the side chains. So I will go ahead and circle all the side chains. Or kind of box them. And then let's go through each. So the ones that would be most likely the least is something that doesn't even have a dipole, and then the strongest one is if we had an ion. So if we look at these guys, choice E doesn't have any polar bonds on it because it's just carbons and hydrogens. So this one would be induced dipole, dipole, so that one's pretty weak. So this one is not going to be true. And then let's look at A next. So A would have dipole-dipole interactions, because with this hydroxyl group, you do have a dipole. So we can think about this one. The same thing with choice B. You do have this electronegative sulfur here. So it's going to be similar to A. And then the same thing with C. We also have a hydroxyl group here. So all of those would be dipole-dipole interactions. However, this one, choice D, is actually charged. So we have a positive cation here. And that is going to have the strongest interaction with water because it's ion dipole. So this one, D, is correct. So next, in a protein, the sequence of amino acids refers to which level of protein structure. So this one is just going to be a memorization problem. Either you know it or you don't. So in this one, I will just go through what all these mean. So primary structure is the order of amino acids. That should be an O. So we know that this one is going to be A, but we'll go through the rest just for your knowledge. So secondary structure refers to the hydrogen bonding of the amino acids. So, and that will show you whether you have some patterns that we call motifs called alpha helices and beta sheets. And then tertiary structure talks about the side chains. So for number 2, the side chains don't matter except for a few very, very specific cases, but the side chains are involved in the tertiary structure because that gives you the total shape of the protein. And then, sorry, not D, number 4. Quaternary structure is not always observed, but it refers to protein-protein interactions. So if you have something like hemoglobin, which has 4 subunits, so for hemoglobin, it's shaped kind of like this ring, which I didn't draw very well, but here, I'll erase that. So it kind of has two bigger subunits and then 2 smaller ones. So these interactions are going to be tertiary or quaternary structure because we're interacting different actual parts of the protein which are totally separate from each other and aren't connected. Their chains aren't connected at all. So they're almost like separate proteins. So for this one, it just asks which is just the sequence of amino acids. So that's going to be primary. So this image represents which level of protein structure. So if we go back to our list, it's not just amino acid order because we have shapes. It's not going to be just H bonding because we have more than just these alpha helices, and then these yellow lines are supposed to be beta sheets. But we see the entire shape of the protein. So that's at least going to be tertiary. But our question is, do we see any evidence of quaternary structure? So that's going to be if you have multiple parts of the protein interacting. And there's nothing here that shows us that the chain stops and becomes a different domain or a different chain of the protein. So we're going to say that this one is going to be tertiary because we don't see any evidence of quaternary structure here. So in question 7, it asks, in an alpha helix, which direction do the side chains of the amino acid point? So alpha helices are something that is observed in the secondary structure, and it doesn't involve the side chains. So alpha helices are very tight turns, basically as tight as this molecule can allow. So if you think about this logically, if alpha helices don't involve any bonding between the side chains, then they're probably not going to be in the middle of the alpha helix because there's just not room for them. They would have to be interacting very tightly. So the only place that they can really go is they could be on the outside, and they can be on both sides. And what we'll find with proteins is sometimes you might have a protein that is, let's say it's in the cell membrane. And this alpha helix might have one side that's facing the water of the cell, and one side is facing the lipid membrane, so a really nonpolar environment where this side is polar. So the side chains of amino acids which are on the nonpolar side will probably be nonpolar protein or nonpolar amino acids, and then on this other side they will probably be the polar amino acids. So the answer to this one is going to be on the exterior of the helix. So which of the following intermolecular forces or structural elements has an influence on tertiary protein structure? So just to call back what tertiary structure is, that is the interaction of the side chains. So we have different side chains in our amino acids. They can be really nonpolar, like this is just a carbon chain. This is very nonpolar, that's isoleucine. Or we can have very polar ones where we have like hydroxyl groups, or we can even have sulfur groups, which these like to form what we call disulfide bonds. This is called cysteine. And we can have 2 of these from different parts of the molecule or different parts of the protein actually react together and bond with each other. So then if we have a protein which looks like this is a very realistic sketch of a protein. We can have 2 parts of the protein be held together by these disulfide bonds, so it stabilizes it. So we can have hydrogen bonding because we can have this H bond with another one somewhere else in the protein. We can have disulfide bonds, which we already talked about. We can have dispersion interactions. That's kind of like the London dispersion or van der Waals forces with the carbon chain groups, which there are a lot of. And then we can also have ion dipole interactions because we might have charged proteins or charged amino acid side chains like lysine, like this one, and that can interact with dipoles in other parts of the protein. So the answer to this will be all of the above. So this is just going to be a memorization question. Of the nucleobases listed below, which one is not found in DNA? So DNA has, A, T, C, and G. And then RNA has, RNA has A, U, C, and G. So the one that's not found in DNA is U, uracil. And then which of the following is the correct pairing? So adenine pairs with thymine and cytosine pairs with guanine. Adenine can also pair with uracil in RNA. So we will draw this. So let's go through our answer choices. Adenine and thymine, well, that one's right. That didn't last long. Cytidine and thymine. Cytidine is not actually the correct form of a nucleic acid. It's cytosine. So this one is incorrect because C does not pair with T. Thymine and guanine don't pair with each other because guanine pairs with cytosine. Adenine and guanine don't pair together. And then cytidine and adenine, of course that's wrong because adenine pairs with thymine and cytosine pairs with guanine. Okay, so here is a picture of guanine. And this question asks how many sigma and how many pi bonds are present. So we're just going to count all of these up. But first, I want to add in any hydrogens that aren't shown yet, because we'll have to count the bonds with hydrogen in our sigma bonds. So all the carbons have to have 4 bonds, and the nitrogens have to have 3. So fortunately for us, the nitrogens already have their hydrogens shown, and then this oxygen doesn't have any because it already has 2 bonds. But the carbons aren't all shown. So this carbon already has 4 because we've got 1, 2, 3, 4. This carbon already has 4. This carbon already has 4. This one has 4, but this one right here doesn't. So let's go ahead and count. So first I'm just going to I'm going to count the skeleton, the carbon bonds for the sigma bonds, and then I will add the hydrogen ones. So here, I'll start here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So now let's add the hydrogen ones. 14, 15, 16, 17. So we have 17 sigma bonds. That's the hard part. And then we'll count the pi bonds. That's just any double bonds that we have. So we've got 1 here, 1 here, 1 here. So that's going to be, let's see, 1, 2, oh, I didn't count this one. So 1, 2, 3, 4. So we have 4 pi bonds. So the answer is going to be E. All right. Which of the following biomolecules comprises most of the cell membrane? So this one is just going to be a knowledge-based question, but the cell membrane is made up of what are called phospholipids. So as you can tell from the name, phospholipids are a polar head group with a nonpolar fatty acid chain tail, and a whole bunch of these make up the membrane in what we call a phospholipid bilayer, because it's a 2-part layer. It has 2 layers that are kind of opposite each other. So we have polar head groups, which interact with the water, which are on these two sides of the membrane, so outside the cell and inside the cell. And then we have these non-polar tails toward the center. And this makes it so that water or polar molecules have a pretty tough time crossing the membrane. So that helps to keep the things out of the cell that we don't want and keep the things in that need to stay in. So that's why the answer to this one is going to be C, lipids. So now we'll talk about what is the repeating unit of DNA. If you'll remember back from the polymers, this is just a knowledge-based question too. It's going to be answer choice A, because we need to have all three of these things for every monomer unit, because our chain is made up of our sugar, and then we have a phosphate group, which is negatively charged, and then we have our base. Which will be right here. So the repeating unit is going to be all three of those things. So phosphate, sugar, and then a base. So the next question. Doxorubicin is an anti-cancer drug that interrupts DNA copying during cell replication. So what functional group is not present in doxorubicin? So let's go through each of these and see if we can find them. So A is alcohol. So an alcohol is an OH group. And it looks like we found it right here. There's a lot of them, but here's a couple. And then the next one is an amine. So amines are when you have a nitrogen, you might have H2, just one H. No H's, still counts as an amine. So that one is found here. An ether is when you have a carbon group, so like we call that an R group with an oxygen and then also bound to another one. And this one is going to be found here and also here. Carboxylic acid. It's a very common functional group, even though it seems pretty specific, but we have a double bonded O to a carbon with an OH. So let's find our double bonded O's and see if any of those have this hydroxyl group or alcohol group coming off of them. So here we've got a double bonded O, but we don't have an OH one step away from it. Same thing with this one. We don't have any OHs that are super close. No double bonded O's over here. We have one right here, and this kind of looks like a carboxylic acid, but this hydroxyl group has to be connected to the same carbon that is the carbonyl, so the double bonded O. So this one, the hydroxyl group, is one step away, what we call the beta carbon. So it's not on the carbonyl carbon. So this one is not a carboxylic acid. So the answer is going to be D. But let's look for some ketones to make sure that's not the answer. So a ketone is a carbonyl that is found in the center or like the middle of a molecule with R groups on either side, so carbons on either side. That can be distinguished from the other kind of carbonyl group that we know about, which is an aldehyde, which just means that it's a carbonyl that's on the end of a molecule because it has an H on one side and then carbons on the other side. So we can find some ketones because this one's a ketone, this one is a ketone, this guy is a ketone, and I think that's it. So the answer here is going to be carboxylic acid. All right, which of the following acids is most like the acidic part of RNA and DNA? So if I come back here and look at the repeating unit of DNA, we can see that this PO4, this phosphate group, has a negative charge on it, which means that it can act as an acid because it could accept a hydrogen here, or it could give away a hydrogen because we have this negative charge. So let's look at our answer choices here. A is sulfuric acid. We don't have any sulfur in our DNA. Phosphoric acid, which is H3PO4, sounds pretty promising because it looks just like very, very similar to our phosphate. So this will have H's on it in this form, but if you put it into water or something, then we can lose our H's and we can have you can lose just one, or you can lose lots of H's depending on how acidic or the pH of your solution is. There's one negative there. So this one looks like a potential. We have nitric acid, which your nitrogenous base does have nitrogen on it, but it doesn't have what we call nitro groups, which have oxygen on them. So that one's not going to be correct. And, which by the way, this is NO2. And then hydrochloric acid, there is no chlorine in DNA. And then carboxylic acid, we don't actually have any carboxylic acids in DNA. We have hydroxyl groups, but we don't have any carboxylic acids. So that one might seem like one that you could pick because it's found so often in the biomolecules, but it's not found in DNA. So the answer here is going to be phosphoric acid. Okay, so what DNA sequence is complementary to the sequence shown here? So if we remember our base pairing rules, A goes with T and C goes with G. So I'm going to draw this bigger so that we can pair it. So it's going to be ACG, GAT, AT, CAGA. And now we'll do the complementary strand. So A base pairs with T, C goes with G, G goes with C, C, T, A, T, A, G, T, C, T. So if we look at our answer choices, TGCC is what we start with, TGCC, TAT, TAT, AGTCT, AGTCT. So it's going to be answer choice E. Sorry, lots of letters. It's like a tongue twister. So seen below is a Hayworth projection of the carbohydrate glucose. In this projection, the ring is drawn in a plane. So basically it's drawn flat. But what is the hybridization and the real bond angles of the indicated carbon? So what this is asking is, even though this is drawn flat, is it really flat? So we need to look at the hybridization of this carbon to determine if that's true. So this carbon doesn't have any double bonds. So normally a double bond, we take away some p character. So with our orbitals, you have the 1s, or excuse me, the 2s, and then you have the 2p up here, which has 3 orbital, 3 locations that we can put electrons in. So if carbon has 4, it likes to split evenly with all of the orbitals that it can use. So if we don't have any double bonds, then all these kind of mix together. We call it hybridization, so that they're all equal. But if we have a pi bond, then that is only going to be one of the p orbitals, so we basically can't use it, and we just have to mix the rest of them, so the rest of them hybridize together. But at this carbon, we have 1, 2, 3, 4 bonds, and they're all sigma bonds. So it's just going to be an S and 3 P's that can mix together. So all of these with 1 electron in each of them is going to be called the sp3 orbitals. So this is going to be an sp3 carbon. And then if we remember from the bond angle lab, if you have 4 things on a carbon and none of them are lone pairs, even if some of them are lone pairs, your angle is going to be 109.5 between each thing. This is what we call tetrahedral. So the answer is going to be sp3 and 109.5 degrees. So that's going to be answer choice D. All right, and I think that was the last question, so that wraps up this week's PARTE Packet.